As you are all getting settled, I would like to draw a little attention to that this is the last in our series of highlighting the BRI term chair holders. So Dr. Churchland is the holder of the Arnold Scheibel Chair in Neuroscience. And I also just want to remember Arnie Scheibel just with a few words in that he was one of the pioneers here at UCLA in doing research, in doing neuroscience research. He came very early on in the existence of UCLA, worked at UCLA in the School of Medicine until he passed away, pretty much until the last or second to last year before he passed away. He donated 3 term chairs to UCLA and also has a really large involvement in the undergraduate neuroscience community in giving now for the 3rd year funding for undergraduates to do research in the lab. So those are the Arnold Scheibel scholars. Arnie was as I sort of indicated, he was a really gifted teacher, mentor to many students. He was teaching over many years the neuroanatomy class to undergraduates. And I still remember talking to one of the undergraduates, and he said, yes, you know, Arnold Scheibel, he's just the greatest, Dr. Scheibel, you know, he rocks. And then I asked him what he got in the class, and he actually failed the class with a D. And he was still super excited about Dr. Scheibel, sort of just showing you how dedicated a mentor he was. He also did some pioneering research on the reticular system in the spinal cord, trying to figure out the function related to the anatomy of the spinal cord. So that gets us to today's speaker, Dr. Churchland. A lot of you know her, so I don't really want to spend a lot of time introducing her. But together with Arnold Scheibel, she really shares a dedication to mentoring, a dedication to making neuroscience accessible for everybody at all levels, and really trying to engage people of diverse and different backgrounds. So I think the term chair is really appropriate. Dr. Churchland did her undergraduate degree on the East Coast at the Wellesley College. Thank you. That there was just a blank in my mind. Then moved to UCSF and worked with Steve Lisberger, and then did her postdoctoral work with, and I'm again blanking with Mike Shadlin. Thank you. I should have brought my notes, but that's one of the things that actually was left behind in my office. And she then moved to Cold Spring Harbor, where she started her own lab, moved through the ranks at Cold Spring Harbor. And in 2020, we were very lucky to attract her to UCLA, where she's been ever since the COVID started. Saw herself through the COVID, and now at the end of the COVID, we finally get to hear from her directly. So, Anne, welcome. Thank you. Oh, and I also, before I fully give over, I want to apologize to Anne and to everybody who's in the virtual environment for the technical difficulties we're having in this room in sharing both things on Zoom and in reality. And so Dr. Churchland actually doesn't have access to all of her notes. There's no sound working, but hopefully we can still work through that. So sorry. And
okay, that's okay. I think I'm going to pass on that microphone, not a microscope, because I have my own. How's the sound? Can everyone hear me okay? All right, thank you so much for that lovely introduction. And indeed, I have a hunch there's going to be some bizarre technical glitches that happen over the course of the talk. And I think rather than feel, you know, burdened by these, we should take a lighthearted approach. Maybe someone could make a bingo card with spaces for, you know, Zoom allowances, or if I get an annoying message, complaining message from one of my kids that they screen, then that's 10 points for that. So we'll try to take a lighthearted approach and just see how it goes. So I'm really honored to be giving this lecture today. I didn't have the pleasure of knowing Dr. Scheibel, but I'm really honored to have a chair in his name. And one thing I know about him is that he was really dedicated to his students and to supporting students really generally. So I want to extend a special welcome to all the students in the audience today in his honor. And I wonder if perhaps we could reserve the first question at the end of the seminar for a student, Felix, if that's okay. I think he would think that was just fine. Okay, all right, we got a pop up already. Okay, so let's see if I can advance the slides. No, okay. There we go, amazing, okay. All right, good. So the starting point for the work that I'm gonna tell you about is some experiments that we did a little while ago that have been published already. So I'm gonna go through them rather briefly and then tell you explain how these were kind of a jumping off point for a rather different approach that we've been taking to try to understand the neural activity that supports perceptual decisions. So in my lab, those are the kinds of decisions that we're interested in. And to us, a perceptual decision means a decision where the sensory signals are coming in. They might be a little bit ambiguous or hard to figure out. And the job of the observer is to make a decision about those incoming sensory signals and then to report that decision with the movement. So we had been studying decision making in my lab for a while, and we had focused a lot on certain kinds of movements, which are what the animals and indeed our human subjects use to report a decision. And mostly we and others in the field had focused on what you might call task-aligned instructed movements. So these are movements that you train an animal to do or maybe instruct a human subject to make. So for instance, in the work I'll tell you about today, we train animals to make a licking movement to one of two spouts. And that licking movement might occur in a very stereotyped or task-aligned way on each of three or many more example trials. So people have been thinking about instructed movements for quite a long time, but we started to wonder whether it might be worth also thinking about uninstructed movements. And so these are movements that we didn't ever train the human or animal to make. They make for reasons that are yet unknown. And these movements may or may not occur at a stereotyped time relative to task events. They could either be task aligned or also task independent. And so we were interested in these kinds of movements because we wanted to answer this larger question, which is, do brains prioritize cognition and instructed movements, or do other signals dominate? So to get at this, we developed a spatial localization decision task for mice, where on each trial, the mouse is presented with either an auditory or visual stimulus, and it's either on the left or on the right. And the job of the mouse is simply to detect which side of the mouse the stimulus is, and to make a licking movement to the corresponding side. And I should say that this version of the task is a pretty simple one. I'll tell you about a more complex version later on. And it builds on a growing body of work coming from my lab over the last, I guess, 11 years now, where we've been trying to study, to borrow behaviors from the human perceptual and also non-human primate literature, and then port them to rodents where we have pretty good access to neural circuits. So I'm going to show you what the behavior looks like. This is, you're going to see a video of the mouse. And from this point of view, the camera is underneath the mouse. So you're kind of seeing the mouse from the bottom up, and that's its on a little nose at the top of the screen. And I'm going to show you what happens when the mouse initiates a trial. There are two little handles at the bottom that move in. And by making contact with those handles, the mouse indicates to us that it's ready to initiate a trial. This is now the mouse from the opposite point of view. You're seeing it from the back. That's its little tail hanging down there at the bottom of the screen. And this is a visual trial with the stimulus on the right. So hopefully you can tell that there's a visual stimulus on the right. And if you were a mouse in the task, you'd have to keep that in mind for a 1 second delay period. And after that, it would be time to make a response. So this is the same view that you saw a moment ago, but now you can look at the top of the screen, and those two little lick spouts are going to move in, and the animal will make a choice. And then the spout that the animal didn't choose, the one on the right, that moves out of the way just to prevent the animal from changing its mind. So remember that our big question was to understand whether in trained experts, whether task and decision signals are dominated in the brain or whether other signals dominate the neural activity. So in going about this, we weren't totally sure which brain area would be the right one to look at. So we wanted to take kind of a more open-minded approach than our usual approach, where we focus in on one area, and to look at as much of the dorsal cortex as we possibly could. And so to do this, we developed a technique in the lab that's now becoming rather popular called wide field imaging. And the way that works is as follows. So we start with the mouse that is expressing genetically encoded calcium indicators in all of its excitatory neurons. So remember that for later, because later on we're going to go a little more granular. But in these experiments, we're measuring all excitatory neurons together. And what that means is that when a neuron fires, it turns green, and we can capture the photons from that neuron with a camera at the top of this custom-built wide field macroscope that we construct in the lab. For any aficionados in the lab, you'll note that we actually illuminate the GCamp with two different wavelengths. This one to directly activate the GCamp, and this one is a shorter, this is a more violet light, shorter wavelength that we use for hemodynamic correction. And if you're interested in testing out this approach, we found it to be pretty useful, and people were emailing us a lot asking how we did it. So we actually wrote a protocol on this that was led by João Cucho, who's a postdoc in my lab. So if you're interested in it, check it out, and maybe you could build one yourself. It's much easier and also cheaper to build than a 2-photon setup. So if you're just getting started with imaging, it can be a good way to go. Okay, so having built this setup and developed this behavior, now we were in a position to get at this question I told you about a moment ago about movements and task signals dominating. So I'm going to show you now the data that it looked like to us as we were watching it come in for the first time. This is a fairly raw version of the data. I'll show you simultaneously the calcium signals from the brain. That's what you see on the right, and also the video of the mouse on the left. This is the what we call the body cam. And these are slowed down quite a bit so that you can see what's happening. So you can see there's lots of fluctuations there. I stopped the movie here to point out the first of two things that kind of jumped out at us. And the first thing is that the activity was pretty widespread. So there's kind of red pixels all across the brain. And if you continue to watch as the trial kind of unfolds, you'll see that that really remains true. There's activity really all over the place, especially around the time of the movement, even though this seemed to us like a pretty simple task, right? Just like lights on the right, lick to the right. It wasn't totally obvious to us that so many different neural structures would be recruited for such a relatively simple task. The other thing that was surprising to us is something that you'll need to look at 4 trials at the same time to understand. So now this is the same kind of similar to the slide you saw a moment ago, but now here are 4 such trials that you can view simultaneously. And I'll play these all at the same time. And what jumped out at us when we were watching these videos is that even though the mouse is doing something pretty similar from trial to trial, at least ostensibly, the pattern of neural activity is very different across the 4 trials. And then so that was the second thing that jumped out at us. The first, remember, was that the activity was widespread. And the second was that it was really variable trial to trial. Okay, so in order to understand these 2 mysterious features of the data, we decided that we wanted to build an encoding model where we could try to understand what was driving these single trial fluctuations in all of the widespread areas that we observed. So we took the data and shrunk it down a little bit. And then our goal here was for every pixel in the brain, I've just picked an example one here, we wanted to understand and ultimately be able to predict using held out data all of the single trial fluctuations. So like if this is trial 1, trial 2, and so on, we wanted to be able to predict all of those little wiggles and bumps that you see on individual trials. So to get at that, we decided to take advantage of first the many behavioral variables that we had, and we wondered whether the presence or absence or timing of those variables might be relevant to understanding those fluctuations. And indeed, these occur at different moments on every trial, so that seemed a reasonable candidate. And then remember that we had also taken video in conjunction with this decision-making behavior. So we actually just took all of the video and squished it down to the top 200 PCs, which I've shown you here. And they just look like really kind of random squiggles, because that's what happens when you do PCA. You get sort of strange combinations of things. And we decided, well, why don't we just take all of these variables together? We're going to put them all in a big design matrix, and then we'll just use L2 regression to find the weights that best map all of the behavioral variables to the neural activity. So then once we have those weights, then we can take held out trials that we didn't train the model on, and we can see how well we can predict their activity. So the 3 trials on the right are held out trials, and I'm going to now overlay in red the prediction of the model. So like it was kind of good. I was pleasantly surprised. We were able to capture a lot of these bumps and wiggles that were present in the single trial activity. And indeed, when we looked across the entire brain, we found that we were able to predict the activity pretty well. So the quantity that I've plotted here is CVR squared. That stands for cross-validated R squared, just the correlation. So it's really simple. We just take the cross-validated, the prediction from the cross-validated trials, and then we correlate that with like what actually happened in the data, and that's the CVR squared. So overall, we were able to explain about 42% of single trial variance in the measurements that we took. So this was kind of cool. And now this allowed us to kind of get at this question that I've been highlighting throughout the talk: which signals are the important ones? And there's really 2 kinds of things that you can learn from this sort of a model. So what I've shown you here is just basically that the model works. That's great. But what can we really learn from the model? Well, the first thing is that we can look at the temporal event kernels that are learned by the model. That's basically the time varying weight patterns that the model is learning when we train it. And those temporal event kernels that are learned by the model, they link brain activity with specific decision variables, because of course we have separate weights for each of the variables, which I mentioned a moment ago. So for example, if we look at the kernel that was learned for the visual stimulus, this is unfolded over space, and you can see that there's lots of weighting in area V1 at the back of the brain. This is for a rightward stimulus, so it makes sense that the weights are high in left V1 on the contralateral side. So all this means is that, those are the places in the brain that explain neural activity from that stimulus. And then we can, these two visualizations are for during and after the stimulus. And if we focus in on one particular part of the brain, in this case V1, and then unfold over time, then you can see these temporal event kernels here. And you can see that the model can learn pretty complex temporal dynamics. And in the case of the visual stimulus in area V1, this is mostly confirmatory, right? This sort of has the expected temporal dynamics that we would have guessed based on the well-established response properties of V1. Okay, cool. So here's another example. This is one which is now an instructed movement. Remember, those are ones that we taught the animal to make. This is for the handle grab. And if we then again unfold in this kind of somatosensory motor region in the cortex, you can see that there's activity that's peaking a little bit before and then also during the movement. And then finally, this is the event kernel for sniffing. So this is an uninstructed movement. We don't tell the animals to sniff, but they like to do that anyway. And you can see that there's lots of heavily suppression and then weighting in the olfactory bulb. Has a kind of interesting complex temporal dynamics that you can see down here. So by looking at these event kernels, we can learn about the pattern by which these different task variables or instructed or uninstructed variables, how they impact the neural activity. But that's only part of the story, right? We don't just want to know kind of how and where these variables affect neural activity. We also want to know how much, right? What's the magnitude of each of those variables? And remember, that was kind of the original question that we set out to answer. So to get at the magnitude of the correlation, that's that CVR squared, between the model's prediction and the data, uncover which variables dominate. So we can look at that value, the correlation or CVR squared for all the variables in the model. And here's a bunch that I've grouped together and called task variables. And you can see that they account for some amount of variance. So that's good. And here's the same visualization for instructed movements. People have believed for a long time that these instructed movements have a big impact on neural activity, and they are right because they account for quite a lot of variance. But the really surprising part, the surprising finding for us was that the largest impact, the variables that explain most of the variance, were uninstructed movements. So movements that we never even taught the animal to make. And we were originally quite skeptical of this conclusion and did many additional analyses to try to really convince ourselves that this was true, including the more conservative delta R squared that you see at the bottom, where we systematically eliminate the variables one at a time from the model and look at how much worse the model performs. It's a much more conservative test. But basically, no matter what kind of test we did, we still came to the same conclusion that these uninstructed movements really dominate neural activity. So if you look at the variables that are in that group, they're pretty diverse, right? Like pupil, hind paw, nose, body, whisking, video, that's just the raw video squished down, and video ME is just the motion energy. That's just the first derivative of that video. And although all of these are lumped together, it would be fair to say that these are not really all movements of the same sort, right? So something like pupil diameter probably explains neural variance because it's informative about neural state, and neural state modulates neural activity very broadly. Whereas movements of the hind paw, for instance, those are more effector specific. Although there can be some interesting bleed through between the two that I'd be happy to tell you about in the discussion. Okay, so we verified these doing a single cell measurement. There's lots of reasons why when you measure wide field activity and it's a pooled signal over many neurons, why you might come to misleading conclusions. So we wanted to verify using a single neuron approach. We did this both with neural pixels and also what you see here with 2-photon imaging, that the same results were true. So we imaged in 5 different neural areas and did automatic segmentation with Sweet2P. We then verified that the time averaged responses, that's those black traces there, were the same as what has been reported in previous areas. And then we did exactly the same analysis, except now instead of trying to predict pixels, we're trying to predict individual single cells. And I'll just tell you just briefly what this plot is. So on the vertical axis, that's this CVR squared again. And then the horizontal axis, that's all of the neurons, and they're all lined up. And they're lined up based on how much variance we could account for in that neuron. So the one where we could account for the most variance is at the left and the least variance is at the right. And that's why it has that kind of nice, like S-shaped, beautiful S-shaped curve there, because we put them in that order. Okay, that's not the result. The result is this. So if you look at the contributions, again, of the different variable groups, uninstructed is black. The main take home is black is almost always at the top. So for almost every single neuron we measured, we accounted for the most variance using uninstructed movements, much more so than task and instructed. For anyone who wants to think a little bit more deeply about this, I think there's a second really interesting point, which is that the distribution of these three variables is quite idiosyncratic across neurons. So maybe in an easier world, if we knew that like 10% of the variance was always, task variables, we could sort of proceed accordingly. But in fact, neurons seem like they reflect really idiosyncratic mixtures of all these variables. They seem to have mixed selectivity that is really quite random. And so if you measure a neuron, it can be really tempting to conclude based on its average firing rate that it might reflect, as I used to conclude, my favorite computation of one sort or another. But I think what this really taught us is that whenever you get those trial average means, you really need to dig deep and understand which variables are driving that mean activity. And is it really only the variable that you think? Okay, all right, so when we sort of stood back to take stock, we felt that there was a lot that was pretty novel in this approach, right? We had this kind of somewhat hypothesis-free or hypothesis-light approach in that we measured neural activity across the entire dorsal cortex and considered many, many areas in our analysis. We also included many more movements than previous studies had used. And we were, in a way, kind of, again, hypothesis-free about that in the sense that we just took the raw video and like put it in the model and let the model have at it, rather than having specific hypotheses about which movements might matter. And that turned out to be a useful approach because we hadn't really anticipated that movements would matter to the extent that they did. So having this kind of hypothesis-free approach was kind of cool and unique about this study. But in a lot of other ways, I think, to be fair, really, this was kind of a twist or a variant on what's really a common approach in systems neuroscience, which is that one measures a whole bunch of neurons together, in this case, all excitatory neurons, and makes the sort of default assumption that they're all part of one group. These are all excitatory neurons. Excitatory neurons are one group. We're measuring them all together. That's kind of a reasonable thing to do. And so looking back, I think this was kind of strange because all the while that we were pooling all these excitatory neurons together and not really thinking about their divisions, there was really like a revolution going on in other parts of neuroscience, which is understanding the importance of cell types in the brain. So this is priority number one of the BRAIN initiative, which is to understand the diversity of cell types. And this is widely acknowledged to be one of the flagship successes of the BRAIN initiative. It's led to many, many papers which define cell types based on transcriptional profile, developmental lineage, sometimes both together. About 3 weeks ago was maybe the biggest week for cell types in the history of the world. There were 17 papers in Nature on the subject, including one big one from the BRAIN Initiative Cell Census Network led by Honghui Zheng at Allen Institute and a number of others. So among systems neuroscientists, these functional differences have been widely recognized for inhibitory neurons, right? We've all read many papers about VIP and PV neurons and SST. And to summarize that really large and very interesting body of work in just one slide, they do different things. They're not all the same. You can't go into the brain and say, I'm measuring inhibitory neurons, and that's the end of it. They do different things. They have completely distinct roles. And those roles differ area to area as well. It's been kind of a whole subfield of neuroscience to understand what these inhibitory neurons do. But for excitatory pyramidal neurons, which are no less diverse, perhaps they're even more diverse depending on how you count, that is much less widely accepted as an important thing to consider when measuring neural activity. And so when we started to think about this, we realized that on the one hand, there's really now, thanks to the BRAIN Initiative and efforts in other countries as well, extensive knowledge of the diversity among excitatory pyramidal neurons. And on the other hand, there's approaches like ours where there's really insufficient consideration of this diversity in experiments that link brain to behavior. And as we thought about this more and more, we realized that this was at the very least a gap, and maybe kind of more like a chasm. And we thought about why that was. And I think there has been or have been a number of papers that have begun to look at the role of excitatory cell types within areas, but there's still a lot that isn't known. And I think we felt like we were in a unique position to kind of get this field moving in a new direction, partly by virtue of the fact that using wide field imaging, we're able to look at the brain-wide activity patterns of different cell types all at the same time. So we can not only zoom in and look at individual areas and understand whether different excitatory cell types play a role, we can also look at their cortex-wide activity patterns and see if those differ as well. Okay, so I'm going to tell you about a couple of cell types that we studied. How am I doing on time? I have no clock or anything. Okay. 12:97, amazing. Okay. Okay, so the first class that we focused on is called a FESF2. It's a transcription factor that plays a big role in development. I'm going to refer to these cells in the talk as PT neurons. That stands for pyramidal tract, and they're called that based on where they project, which you'll see in a moment. Importantly, we acquired mice that expressed calcium indicators exclusively in FESF neurons through a collaboration with my former colleague from Cold Spring Harbor, Josh Wong, who's now at Duke. And he provided us with these mice. And here you can see mice in a, sorry, not mice in a FESF neuron, neurons in a FESF mouse. So I'm going to draw your attention here. Every time there's a bright white spot, that means there's a FESF neuron there. You can see they're mostly pretty deep in the brain. They're in layer 5B. And as we kind of take a movie that goes all the way through the brain from the back to the front, you can see that they're really very evenly distributed, making this quite well suited for wide field imaging. They're all the same, always in layer 5B, no matter where you are in the brain. And they're called PT or pyramidal tract because they mostly project down. So they have a small cortical projection, but mostly project to the pons, the superior colliculus, the cortical spinal tract, and so on. The second class that we considered were plexin D1, which is another developmental regulator. And I'll refer to these as IT or intratelencephalic neurons, noting that actually IT neurons are a rather broad group. And that's a topic we can return to in the discussion if folks are interested. These neurons are a little complicated, and this will come back to haunt us later in the talk. They're located in two different layers, in layer 4 and then also in layer 5A. Sorry, in layer 2-3 and layer 5A. Here's the same movie. You can see they're present, albeit slightly less homogeneously as you go from the back to the front of the brain. These neurons have a really different projection target. They project both cortically and also corticostriatally. So both the cortex on the other side and also the striatum, making them quite distinct from FESF neurons. So these are the two cell types that were the focus of our study, and we decided that we would compare their activity patterns to EMX mice, in which we measure all of the excitatory neurons together. It's not totally clear how one should think about what I'll refer to as EMX mice, because they're in some ways a superset of PT and IT. But remember that there are lots of neurons in the brain that are not labeled by our PT and IT labels. They might be intratelencephalic, but not be plexin D1. They might be projecting to the thalamus, which wouldn't be labeled by either of these. So EMX is sort of like a superset plus. It's like a superset with a whole bunch of other stuff in it, perhaps dominated by more superficial signals. And I'll return to that point. So I'll hope to answer just two questions about this new work, which is emerging. We have a paper on the bioRxiv, but it's unpublished. First, do PT and IT cells exhibit unique cortical activity patterns? And second, do PT and IT cells play distinct functional roles during decision making? So I'll start there. This is histology to verify that we have the labels in the right place. We also always take retinotopic maps with all of our animals to make sure that we can measure reasonable signals. Having the ability to measure these maps through the skull is a really important verification that we're measuring reasonable signals at all, which was not in no way a foregone conclusion, especially for those PT neurons, which remember are all the way down in layer 5B. But amazingly, we were able to get good retinotopic maps, which was reassuring. I'll just show you now a couple of movies that are aligned to decision-making events taken in separate mice for PT neurons and IT neurons. And the main thing I want to give you is here is kind of a bird's eye view of what these activity patterns look like, and to start to judge for yourself whether it seems like the activity patterns across the cortex are similar or not so similar in PT and IT neurons.
Not similar.
So yeah, maybe not that similar. That was what really jumped out at us. And when we first started to just look at this data coming in and to see how different these patterns of activity were, we realized that we needed a way to quantify this, to be able to look at cortex-wide patterns, compare them to each other, both in terms of the spatial layout of the patterns and also in terms of their complexity. But you can imagine this is a little bit of a daunting task, right? Because what do we have at the end of the day? We just have this huge stack of images for each of these three cell types. It's not totally obvious how one will compare them. So the first thing that we did was to look at how complex they were by taking all of the data and using semi-non-negative matrix factorization, or SNMF, to decompose each big set of data into spatial and temporal components, allowing us to kind of compare separately the spatial and temporal pieces. So then we can ask, how many components do we need for each cell type to account for 99% of the total variance? And here's the data here. So the labels are on the horizontal axis there. So let me just walk you through what this is. So each cell type is plotted separately, and that vertical axis is how many components you need. And then within each cell type, say EMX, each open black circle or open white circle with the black around it is a single mouse. And then for each mouse, we have many, many sessions. So you can see that across mice and even across sessions, it's pretty consistent for EMX and IT. And that we have many more components for those than we do for PT neurons. So what does that mean? That means that the overall spatiotemporal patterns in the PT mice are much simpler. We need fewer spatial and temporal components to account for all of the activity than we do for EMX or IT neurons. So that tells us about how complex the patterns are, but it doesn't really tell us whether they're similar or not. So to get at whether they were similar or not, we took the spatial components for each of the spatial components generated within each mouse line. And we embedded these in a two-dimensional space so that we could start to compare them. And we did that using UMAP, which is a nonlinear embedding technique. So this is what the data looks like. So you can see these three example spatial components from the previous slide. They each correspond to one point on this big UMAP plot. And the reason there are so many points is because, remember, we have a lot of mice and a lot of sessions, and each cell type is colored using the same color scheme as before. And I'll add those labels on in a moment. So what jumped out to us at first is that these spatial components tend to cluster together. So there's a big clump of blue together, big clump of green, big clump of red. That tells us that the different spatial components tend to look like each other within a particular cell type, but they're different from the other cell type. One thing that gives me reassurance about these groupings, these kind of clumps in the two-dimensional space, is that, remember, this is pooling over a lot of mice and also a lot of sessions. So sometimes when you do things like SNMF and you're like pulling a matrix apart based on its variance, you can get kind of weird, slightly idiosyncratic day-to-day changes. Like, that's a reality. But the fact that we see these clumps, despite the fact that many days and many animals are included in this analysis, gives us reassurance that these clumps correspond to real groupings of spatial components. Okay, but we worried here. Okay, so remember, what defines a spatial component to begin with? Well, basically, it's like, okay, you have a little bit of a bright patch here and then a little bit of a dark patch here. That's like what a spatial component is. But what if the reason that we see these differences across the cell types is that there are differences in expression levels? So that despite our best efforts, there tended to be kind of, like more IT cells laterally and more PT cells medially or something like that. So to get at that, we needed a new tool, or a new variant of NMF. So we had a collaboration for a while with Shreya Saxena, who has her own lab now and did a collaboration with us when she was a postdoc in Liam Paninsky's lab. And she developed this kind of cool technique called loca NMF. So for this one, it's the same kind of breaking apart of the matrix, but you seed the analysis with a spatial region. And we did this based on the Allen Institute CCF, kind of a merged version of the Allen Institute CCF to make it a slightly bit less granular than it normally would be. And then by seeding the region with these areas from the Allen Institute CCF, then you tend to have spatial components that are mostly restricted to that area. And as the user, you set a parameter defining how strictly they're contained, and you can kind of play around with that. So here's a couple of examples of the spatial components now from loca NMF, and you can see that they're kind of localized just where they should be. So the key analysis here that was reassuring is that when we looked at the number of components from loca NMF, they were the same for all three cell types. And the reason that this is reassuring is because if there were some gaps, if there were some areas where there just wasn't label, then there would be fewer components in loca NMF when we look at this plot here. So this tells us that the differing complexity that we observed when we looked at whole brain activity is not due to the fact that there are gaps in expression by virtue of the fact that we see similar number of components with loca NMF. So now we can do the same thing and look at the components again in this two-dimensional space, just like we did before. This looks kind of weird compared to the other one, right? It has all these weird patches, but those patches, that's exactly what you're supposed to happen. So like, for example, this patch corresponds to that component. This one is that one, and that one is over there. And all this is telling us is that loca NMF is working, right? Because you have components that tend to be clumped to a particular part of space. Great, that's not news. But we can look inside these patches and look and see whether there's the same clustering we observed in the previous analysis. And here we found really two things. The first thing is that indeed there are clusters. So this is kind of a mostly PT cluster. This is a mostly IT cluster here. And that led us to a conclusion that you can't see at the bottom, which is that PYN types exhibit unique patterns, not only at the macro scale that I told you about a few slides ago, but also at this smaller mesoscale where we're only looking at activity restricted to a particular component. A slight side point for any aficionados in the audience, you'll notice that there are some regions here that are a little less specific. We call these non-specific regions. And when we looked at the size of the components compared corresponding to specific versus non-specific regions, we found that the regions corresponding to the specific clusters tended to be smaller, and that was overall true. So this is kind of a side point, but I think it's sort of neat because it suggests that beyond the structure of the Allen Institute, there might be kind of an interesting functionally defined substructure where you have kind of smaller sub areas or mini areas dominated by one cell type, and then some larger kind of super areas that have mixed cell types, suggesting kind of an additional structural organization beyond what you can see with the normal Allen CCF. So we're still trying to understand what exactly these areas mean, but it was kind of neat to observe that in the data. Okay, so far I've just told you about large scale and mesoscale activity patterns, leaving aside decision making for a moment. Let's get back to decision making. Do PT and IT cells play distinct functional roles? We used roughly the same task as before, except that this time it's a discrimination version where there are stimuli on both sides. This is only auditory for this part. And the job of the animal is to report which side has more clicks. These are psychometric functions here. And we looked at the decision-aligned activity just as we did before, found some interesting and kind of intriguing differences across cell types, and decided to dust off our encoding model and to once again look both at the time-varying patterns of the temporal event kernels and also the magnitude of what the different variable groups could explain. Okay, so we dusted that off. So what I'm showing you here is to connect to the previous study, we look at the overall contribution of movement and task variables for each of the three cell types. And so we first of all reproduced our previous result, which is that movements tend to dominate neural activity. And in fact, we found that to be even slightly more true than for PT than for IT neurons. Now, a natural kind of conclusion here is to think that this is in a way a bit intuitive, because PT neurons, after all, what do PT neurons do? They're downward projecting. Their job is to kind of move the body around. And that turns out to be sort of true and sort of not true. And that became clear when we started to look at the event kernels and their locations and shapes for the onset of the stimulus activity. So remember, this is the same kind of analysis before. These are the kernels learned by the model. And I'll show you that for EMX, IT, and PT for three different regions: frontal, parietal, and auditory. And you can see that these are actually kind of overall fairly similar across the cell types. There tends to be a response that is stimulus aligned. I don't know that it's a sensory response, but it tends to occur at a relatively stereotyped moment relative to the stimulus. And this was not super surprising for EMX and PT neurons. After all, presumably there's lots of sensory signals there, but it was unexpected for the PT neurons. Because if they're really there just to move the body around, you wouldn't necessarily expect them to have a stimulus-driven response that really peters out long before the movement occurs. And note that the arrow is pointing to a response in parietal cortex, which, although it certainly plays a role in movements, is not a motor cortex. So more on this in a moment, but this was a first hint that the PT neurons were doing something that we didn't totally expect. And this is a blow up so that you can see there's really complex and intricate patterns, which we're now zooming in to look at with our 2-photon microscope, so we can start to get a single cell perspective. Okay, so we also looked at the event kernel corresponding to the animal's choice, found some interesting activity patterns, but you'll note that these values are really, really small, because remember that choice is a task variable, and it just doesn't account for very much, for much variance at all. So we were sort of skeptical of that. We switched instead to using a decoding model where we try to guess the animal's choice based on the activity. And we found that we were able to do that pretty well for both PT and IT neurons. And then we can again look at the event kernels learned by the model. These turned out to have really kind of complex and slightly bizarre patterns that we're still trying to understand, certainly for IT and PT, for EMX as well. So I'm just going to focus on these two periods here, stimulus and delay, and on two areas, what we'll call parietal cortex and up here frontal cortex. And I can tell you more detailed anatomy about what those refer to, but the frontal cortex is mostly area ALM, and parietal is mostly area AM, A, and a bit of RL. Okay, so then when we looked at the temporal event kernels, again, to provide insight into the time varying signals, we found that PT and EMX tended to increase, but there was a mysterious declining contribution of IT cells to the choice to the choice kernel. So that means they become less and less important for decoding choice as time goes by in the trial. And that was really quite different from PT and EMX. So this told us that PT and IT neurons have different choice-driven activity in M2, but this really brought up a problem. And the problem is this: when we look at this declining contribution to the choice kernel, we're really stuck because we don't know if that declining role of choice in IT neurons is driven by the cortical or more superficial IT neurons or the deeper corticostriatal neurons. Remember that these neurons are located in two places. And this kind of highlights, I think, the challenge of using a developmental lineage approach to define cell types. In the one hand, I think they really are kind of a cohesive group by virtue of having played a common role in development back in the very early life of the mouse. But on the other hand, when you define cell types this way, because they're so high up in the kind of the hierarchy of cell types, they can sometimes be made up of cells that are rather different from each other. And it's not always obvious to know what to make of that. So we didn't know what to make of that. So we just kind of pulled it apart. So we said, okay, let's step away from developmental lineage definition and now define cell types based on anatomy. We're going to label the cells based on their projection to the striatum. Who cares what they did during development? We're going to label in the cortex all of the cells that have their cell bodies in the cortex that project in the striatum. And we tried to target the whole thing. We verified that this worked. We had really nice coverage. And repeated all of the analyses that I've been telling you about, both looking at the modulation, looking at the stimulus kernels, and then finally looking at the choice kernels with the decoding model. And here we found that when we looked at corticostriatal neurons specifically, that declining contribution to the choice decode was even more prominent. So that means there are still open questions about the more superficial neurons, but for sure, those corticostriatal neurons are having a declining role. So we have some hypotheses about that are kind of emerging, and we have some 2-photon data that's maybe helping us to start to understand this a little bit. But for reasons that are still unfolding, it's clear that IT and PT neurons are doing something very different in the frontal cortex. Okay, but just to take stock, so I told you now just a moment ago about frontal cortex being modulated by the animal's choice, and also this rather surprising result that parietal PT neurons are active during decision formation. But it's hard to know what to make of all of this, because when we think about frontal cortex and parietal cortex, we don't necessarily know what their causal role is during this particular decision-making task. And we really wanted to validate that these areas had a causal role at all, so that we could be sure that these interesting cell type specific differences that we were observing were actually relevant. So the first thing that we did was to do optogenetic suppression using S-Gattaker, and we suppressed separately first in the frontal cortex over the whole trial. And here the vertical axis is performance. So you can see frontal suppression really makes performance much worse. We then did parietal suppression. It also made it much worse. And then on this auditory task, we suppressed V1 activity and saw almost no effect at all, because presumably primary visual cortex isn't needed for this auditory discrimination task. So this was really important because it demonstrated the causal relevance of frontal and parietal cortex, which is really the place where we saw these interesting cell type specific effects. We decided then to take this a step further, and this will be the last data that, that I'll tell you about today, and to not only suppress the entire area, but also suppress it in a cell type specific way so that we could start to have a sense of whether the functional role that was hinted at by our measurements was also seen when we manipulated optogenetically. So this is kind of harder. So now we're stimulating in a cell type specific way, and we also did it in a temporally specific way. So we interleaved trials where we suppressed activity at many different moments over the course of the decision. And I'll start by showing you data from suppressing the parietal cortex. So remember EMX, this is when we are suppressing all of the excitatory neurons together. We saw a pretty big impact on decision making. The vertical axis is now impairment. So that's sort of how much worse did we make the behavior based on this stimulation. This is sort of a temporal unfolding of the result I showed you a moment ago. When we suppressed IT and CST neurons, we did see a slight impairment, but it was rather modest. And when we suppressed PT neurons, we saw a really consistent effect that had some really interesting temporal dynamics. So remember, the temptation is to think of PT neurons as being these kind of drivers of movement. But interestingly, the causal role actually declines as you get closer to the movement. So we have a larger causal role when the stimulus is being presented, and really almost no causal role at all during the time of movement preparation and movement execution. So remember, this is in parietal cortex. So this suggests that these PT neurons are doing something in parietal cortex that is probably not directly related to movement, but still has something to do with decision making by virtue of the fact that when we suppress those neurons, we see a worsening of performance. So I think that fits well with the observation that we made before, that PT neurons are also active. They're now not only active, but also causally relevant in the parietal cortex during decision formation. We also repeated these experiments in the frontal cortex and found some frontal dependent responses that are in some ways similar to those that have been reported in previous studies. Okay, so I've told you so far that PT and IT cells drive unique neural dynamics at both a cortex-wide and also a within area scale. That cortical activity and optogenetics reveal distinct functional roles, and that surprisingly, parietal PT neurons had a strong causal role during decision formation. And lastly, that in frontal cortex, all P/Ns were required for accurate choices, but they showed distinct choice tuning, and in particular, this declining contribution to the choice decode from the IT neurons, which was kind of unexpected. Okay, so this work was mostly led by Simon Musil and Richard Sun. Matt Kaufman played a role in the first part of the work in the first part of the talk. None of them are here with me at UCLA because they all went and got their own jobs, although Richard Sun is going to join us next year. And then on your right, you see my lab, who are amazing. Many of them are here today, and I feel really lucky to be working with all of them. I hope in my next talk, I'll be able to report some of their exciting new discoveries, which are unfolding every day before our eyes. And we're also really grateful to all of our funders and to UCLA and to all of you in the audience today. So thanks a lot, and I'd be happy to take any questions.